So my name is Heiland. I'm from Germany, Freiburg. So that is on the border between Switzerland and France, a very nice place. So highly recommend it. If you visit Germany, come by and have a nice view. The city is very similar to Ann Arbor. So we're also like a very small academic city. And so I'm running a group under the Department of Neurosurgery, specifically focusing on high-dimensional data, spatial and single-cell sequencing. And so today we're going to have explore a little bit on one side, the technical part of spatial transcriptomics, how we can deal with these data and what's special about these spatial data. And then on the other hand, we also try to look a little bit on the biological perspective of glioblastoma and the different regions that we can identify in brain tumors. So now it's working. So we are part of a support program from Tenix. So that's a little bit of a disclosure. The topography, or basically the spatial context of glioblastoma and cancer in general, is becoming more and more important since we now have more technologies. But we also noticed it already in the time of Scherer and Virchow, that basically the tumor components and its spatial distribution of different cells, shapes, and morphologies seems to be very important. And so now having these new technologies, like spatial transcriptomics or other like high-dimensional spatially resolved metabolomic techniques, we are now able to explore these differences in the different areas of the tumor and also find interactions and mechanisms that drive these cells. So now in these, I want to go through different topics today, because it should also be a little bit a technical perspective in these kind of seminar series. So we go through challenges and solutions in data analysis and data structure. Then we look how we can use these algorithms or these solutions to have a look on the topography of glioblastomas. We then switch a little bit into more specific immune niches and the immunology of glioblastomas. Finally, look through different areas in the glioblastoma and end up in the necrotic hypoxic core. And then finally, I also might provide some outlooks how potentially we can use spatial transcriptomics in the future as a translational technology. So first of all, I will start with what we have acquired so far. So that is our spatial database that we know using in Freiburg. So there is containing different kinds of tumors, different stages of tumors, and we always trying to collect multi-omic data. So that means we start with the Visium platform for spatial. Then we used MALDI imaging for get an idea of what is the spatially resolved metabolomic landscape of these tumors. We're trying then specifically to zoom in specific areas, and by using imaging mass cytometry, get an idea of the single cell resolution and interactions in different areas. We have also now developed some kind of a new technology, how we can use spatial transcriptomic cDNA libraries to acquire T-cell receptor sequencing data, and then also look on the level of T-cell receptors in space, and then also a single cell data set just basically for reference and also for integration of these data. So these all data come with some kind of problems. Then most of the labs probably noticed that computational analysis is one of the bottlenecks of all these high-dimensional data. So we also try to acquire some and develop some software that can help us to understand the relationships and specifically focus on spatial context of multiple data platforms. So, and this is the three guys that mostly contributed. Here, the upper one is the team that works on the data acquisition, and then here Jan Kuckelhaus, a very talented PhD student who mostly contribute to the software development. So now this one is not. Okay, so when we're talking about software, I also would just like give you a small introduction of what software type we're using. So that is SPATA is a spatial transcriptomic analysis platform that we created. It's basically in R language, but also can be adapted to other languages, or basically we have also here wrappers that we can integrate other languages, especially when it comes to imaging analysis. Python is very important. So this object is basically structured to have imaging data and then transcriptomic, metabolomic, and all other types of data that we can put together in one of a object structure. We can then connect these to single cell data, which is basically very helpful if we're talking about deconvolution. But what we want to provide is always a very tight connection to the imaging data itself, because we believe that in the future it's even more important that we connect these transcriptional data with cell morphology, shapes, or any kind of imaging data that we have. And then finally, we also provide a platform that it is easy to develop different algorithms for post-analysis, especially receptor ligand interaction or neighborhood analysis that can be done. The problem is most of the people do not really have an access to coding and the language. So we also notice that it is very easy to have platforms with direct interfaces for users to have easy access on these data to look in some kind of biological backgrounds and functions. So that brings me to the next problem we're going to talk about, which is data structure and what makes these spatial transcriptomic and spatial data so special. And we first talk about the sparsity of these high dimensional data in general. So that is not only true for spatial transcriptomics, also for single cell data. We're next talking about dependencies in space and hidden dependencies that we have also to consider in our analysis. And finally, then the major problem of deconvolution, because most of these techniques do not have single cell resolution. So we have to infer and deconvolute our single cell data set to find, or at least infer, a single cell resolution. So sparsity is something, in easy words, if you have a lot of zeros in your data set. So, and because we are not sequencing at a certain coverage, we will have a lot of zeros in the data set. And to get rid of these zeros, for example, we can do normalization algorithms in single cell. But the problem in spatial is that if we consider each of these data points as a unique data point, then we might fail because all of these spots are connected to its neighborhood. So what we tried is to figure out if we can use graph representation of the gene expression data and then use autoencoder, which is basically a trained dimensional reduction and then reconstruction to the original data. And these reconstruction data then provide us a little bit a reduction of the sparsity. And finally, we can use these intermediate space, which we call the latent space, then further for clustering and getting some kind of a spatial pattern analysis. When it comes to correlation of these data, also something which is very important to consider, because I see that a lot of times a gene, for example, can be expressed either, for example, if we look at the spot in some kind of a pattern, or it is randomly distributed in the data set. And to measure these, if a gene is expressed in pattern, in clusters, or just randomly distributed, we use something that called autocorrelation. So basically what we correlate is the mean expression of the surrounding spots versus the spot in the middle. And if this is highly correlated, then this gene is not exclusively expressed in one spot, but basically the whole neighborhood expressed the same amount of this gene. And then we call this gene is some kind of in a pattern. So the problem comes when we now want to correlate two genes, because now two genes can be either correlated because in the same distribution, these spots are located in your sample, you have the same amount of expression, or these genes are basically expressed because the spots in the neighborhood expressing the same genes. And so if you want to compare gene expression of one versus another cell, you always have to look at the residuals of your model to check whether these residuals having a high autocorrelation, so being expressed in the same areas or not. And then we can assume from these data analysis, for example, if there is a couple of cells in the neighborhood of these specific cells that we are looking at expressing similar genes or being basically influenced by these cell types at the same time, or basically if there is only a single cell or some kind of a heterogeneity regarding impact of different cell types on each other. So that helps also to get an idea of how this gene expression in space is distributed. So coming to the next problem, which is the cell deconvolution problem. And there's also a lot of algorithms now that providing the idea of which cell types might be at a certain percentage in which of these spots. But one problem is that we don't want to know what cell type is it, but probably we want to know what is the most likely cell of these single cell RNA sequencing data set that is located in one spot. So what we can use is we can use a couple of algorithms, and we can't really use deep learning because we have no ground truth and no gradient descent in this context. So we need to make use of some kind of classical machine learn algorithms. And what we use is, first of all, a convex linear optimization, and then an evolutional algorithms that providing us in combination with the numbers of cells that we definitely have at these spot to really predict some kind of a most likely cell from your single cell reference in your spot. And this is how it would work. For example, here we have a reference data set of 1 million cells from 140 patients, which is most recently published, and then or basically a preprint, and then we trying to integrate the single cell data set in our spatial transcriptomics. And this is how it would look like, for example, if we're just doing a spatial cluster analysis. So if we now integrate these cells, then we can, so for example, here see which is the most likely cell of your single cell data set at which position of your cell, and you can directly transfer the resolution of your spatial transcriptomics data into a single cell data set. But we have also to consider that is inferred, so that is something the computer prints out. That is not real, and it's very hard really to look if this is, so we have no ground truth to really prove that this is real. So now I'm going to talk a little bit about these whole patterns. So we talked already about a pattern that we see in space, and now we want specifically to look if we can identify something like a region that is very similar within our glioblastoma that we can then further characterize, look at this pattern appears across all patients, or if this is something that is only unique for a single patient. And what we have done is basically we first looked at individual spots, and we tried to get rid of spots that have a very high percentage of noise, basically having a lot of different cell types in this spot. So we cleaned these spots up for having a percentage of almost 90% of tumor cells. And then we perform cluster of the spatial transcriptomic data patient-wise. So every patient gets some kind of a number of different clusters, because what we noticed is that if we cluster all patients together, they cluster per patients. And this is also something in single cell data happens more frequently, that basically patients or the tumor cells from each patient has a different genetic background and a different transcriptional profile. You not really can compare them. So after individual clustering of all of these patients, we see gene programs that occur more rapidly across all patients, and other gene programs that were completely unique to a single patient. So then we wanted to know what of these programs have a certain overlap, and we took these overlap and then built up some kind of recurrent spatial signatures that we can see. And then we use these autocorrelations that I already introduced a couple of slides before to test whether these gene programs occur in a distribution across the whole sample, or if it is occurring in some kind of these patterns. And when it comes to a pattern expression, then we said that this is some kind of a spatial transcriptional program that was seen in many patients and specifically mark a region in the tumor. So putting all these together, we see 5 different programs. Basically, one is characterized because it's very similar to what we see in radial glia transcriptional programs. Then we have a reactive immune and a reactive hypoxia here on the bottom program. And then 2 programs that almost covering a OPC-like phenotype and a neurodevelopmental phenotype. And here now we split all these spots into what is the dominant cell phase of these different programs. And what we can see is that basically for a reactive hypoxia and neurodevelopmental program, there is a significant enrichment in G1 dominated spots. And on the other hand, here, this reactive glia program is more dominant in splitting cells and cells or spots that having a dominant expression of mitosis marker. When we look at the histology of these different areas, we could also see that it makes completely sense. These necrotic edge areas that also characterize that as more hypoxic areas, also expressing these hypoxic reactive phenotype. But all the other classical histological areas are not really specific enriched in one of these phenotypes. We then compared this with the literature, and this is basically something that we can just see that there is some kind of a reactive stress phenotype, almost single cell or bulk transcriptomic classifications also show the same. And we have a basic neural-like phenotype and a glia-like phenotype. So this is basically the large different phenotypes that we can identify, and that is across all different signatures that are identified so far. So having now the spatial transcriptomics, and we compare the histological differences here from the leading edge, then the cellular microvascular proliferation, pseudopalisarding until the necrotic areas, we can then also assign our different programs that we have identified to these different regions. And now we're going to just go step down these histological ladder and basically look in these different areas what we can find there. So we start with the leading edge area, and one of the major cell types that we have identified in these leading edge area is reactive astrocytes. So astrocytes basically can transform reactively into an alternative activation and an inflammatory activation. And when we compare some kind of data from other groups that have clearly classified this in like the alternative A2 and inflammatory A1 phenotype, and some kind on another axis of from progenitor state until to mature differentiation, then we can just map in our astrocytes that we found and could see that basically these astrocytes have a very unique phenotype, which is on one side an alternative activation, and on the other hand, they seem to be also developing backwards or differentiated backwards to a more fetal phenotype. So that time we started with this analysis. There was no spatial data at all, but we identified on transcriptional basis some interesting protein markers that could be help us to identify these astrocytes. And one of the markers was PD-L1, a very famous, so CD274, which is highly expressed in these astrocytes. And when we look specifically at different regions of the tumor, we can also see that this comes in these peritumor region. So then a couple of years later, now we have spatial transcriptomics, and now we have a tumor part, a peritumor region, and then here almost normal brain. And when we look where these astrocytes are co-localized, we can again see there was a very nice prediction that time, because they are definitely in the peritumor region and might contribute to immune processes. So we further then want to look at why these specific activation pattern happens. Is this basically on a microglia activation, or is this related on just a cytokine connections in the brain? And what we used is we used a axis cortex that we can gain from glioblastoma surgeries, and we take the non-infiltrated cortex area, and then we also produce a cell line from the core of the tumor. And after 1 day of settle down of these slices, we can re-inject the tumor into the slices and then follow these slices for a couple of days and look at how these astrocytes differentiate. And what we can do is, additionally, we can deplete microglia cells in these slices. So we have a couple of different conditions. One is we have a depleted microglia slide, one with microglia in place, and when we have astrocytes in the slide and astrocytes placed just on the bottom on these co-condition medium. And what we finally find out when we transcriptionally profile these tumors, these astrocytes, is that only astrocytes that are in direct cell contact with microglia cells within the tumor can really have the specific phenotype of progenitor alternative activation. These astrocytes that are just placed on the bottom also showed an alternative activation, but not these fetal differentiation. So we now we go one step further into the cellular parts of the tumor. And when we go into the cellular part, we're switching a little bit from a high density of astrocytes towards a high density of microglia and their macrophages at this point. So we wanted to look specifically in the macrophage on one side, microglia, and also lymphocytes cell types that specifically co-localized in the cellular parts of the tumor. So basically, we did some profiling on single cell, and that time there was a lot of data now coming to characterize the myeloid component. So we focused a little bit more on the T cell component. And so we looked on CD4 and CD8 positive T cells, and what we found is that only the CD8 component of lymphocytes showing up with a dysfunctional exhausted expression phenotype, but this could not be found in the CD4 component of the T cells. Also showing us a little bit that potentially this whole problem of T cell exhaustion might be mainly related to CD8 population. So when we look in these CD8 populations and then make some pseudo-time analysis and trying to differentiate the branches of activation states, we could see that basically these T cells start from a naive state towards a T effector cell state, or they switch into a dysfunctional and finally exhausted phenotype. And when we then look what kinds of cytokine responses are highly correlated to these branches, we could identify that IL-10 and TGF-beta, which is both known as being immune suppressive cytokines, are highly correlated. So now the question is, who is producing these IL-10 and TGF-beta? So who is the reason for the T cells to being exhausted? And there was the need of creating some kind of a new analytic tool. And this time we started to invent these nearest functional connected neighbor analysis, which is basically incorporating spatial transcriptomic data and single cell data, and use these spatial transcriptomic data as anchors to look if a single cell data, or what is the probability of two cells that were identified being related to each other, and what is the actual physical spatial interaction of these cells. And by using these models, some kind of a computational model, we also add the duplicates that we have in our data set, because duplicates basically not only a technical artifact, probably these cells also are just basically physically interacting. And so if we can look at a doublet of myeloid and T cells, so if we end up with our target myeloid cell population and have these more frequently in a doublet with a T cell, another T cell of our target T cells, then it might be a higher physical interaction of these both cells. So it also might be a nice signal that these cells are interacting. So putting all these data together from different models, we finally identified a specific myeloid phenotype, which is HEMOX1 positive CD163 bone-derived macrophages that highly contribute to these T cell exhaustion by IL-10. So if we now inhibit these processes by, for example, experimentally, we could show that a YAG-STAT inhibition of these cells might reverse this progress. And so we tried in a small clinical trial, we used ruxolitinib in a neoadjuvant setting and compared the tissue that we have before and after treatment. And we could definitely see that we enhance the numbers of effector T cells while we reduce the numbers of effector T cells outside of the setting. So that is also a very difficult question. So by looking now in these different areas of the tumor, we noticed myeloid T-cell interaction is a very important thing. And these interaction basically then produce these strong immune suppressive phenotype that we see in brain tumors. So we were interested to see whether these immune reactive phenotype that we identified or the other hypoxic reactive phenotype, which is both having a high percentage of mesenchymal-like differentiated cells, have different amounts of myeloid cells and a different distribution of these populations. And we used IMC to specifically focus in these different areas. And we noticed that, yes, in both, we have a high amount of T cells, or at least we have T cells, and we have myeloid cells. But interestingly, in the hypoxic areas, we identify a lower percentage of myeloid cells. So basically, we thought, potentially, these T cells in these different areas might also be having different behaviors or clonal proliferations. So to look specifically in the question of if there are specific T cells co-localized in hypoxic or myeloid-enriched areas, we then developed a tool, the SPTCR-seq, where we basically used a normal Visium cDNA library and then produced a long-read sequencing data set with nanopore sequencing. And nanopore sequencing then provide us the full transcript that we can then finally use for rearrangement of the T cells. So when we look at these data, and this is, for example, an example of total number of reads from this SPTCR-seq approach compared to just CD3 expression from the spatial transcriptomic data. So from the Visium data, we can see that there's a high similarity, but the amount of reads and details that we get in SPTCR-seq is way stronger. So then we compared clones across different spots, and the question was, is a clone normally only could be identified in a single spot, or can we identify clones that are expanding across multiple spots? And yes, most of the clones can be identified in only a single spot, so basically 80%. But 20% seems to be so proliferative that we can identify the same clone in a couple of different spots. And when we then look how large are these spots away from each other, then basically we have the same length, so the same distance than the normal center to center distance. So basically we can see that they normally not spread more than 2 to 150 micrometer. Another question was, now all these clones are in which area of the tumor that we have identified before. And that was also interesting to see that basically not the reactive immune or reactive hypoxia area is the area that has most of the T clones, but basically the radial glia component of the tumor has in most of the tumors the highest component of T cells. So reactive immune and radial glia have almost 2/3 of all T cell clones that we can identify. So there was also showed us that T cell distribution is very connected to the transcriptional phenotype and cells that we have in the GBM. If we then look at the transcriptional phenotype of each of these clones, and again, then finally look not if a clone is there, but actually which type of a clone and which transcriptional background of this clone is in which area, then we notice that T cells that basically co-localized in or like that occur in these reactive hypoxia areas are mostly memory and effector T cells. But on the other hand, if we look in these reactive immune areas, for example, we see a large percentage of dysfunctional or exhausted T cells. Also showed us that basically these reactive areas can have major differences in T cell responses. Or in other words, there might be a sweet spot in the tumor where you have a clonal proliferation of T cells or a anti-immune reactivity, where the other parts of the tumor are completely exhausted and immune suppressive. So finally, we try to get a little bit better idea of how this T cell, myeloid, and tumor cell interaction works. And so we created a network of all different cellular interactions, receptor ligand interactions that we can have in the tumor. And we basically identified 3 models that are highly co-correlated to clonal proliferation of T cells. So whenever these spots here became big, this model is basically you see more clonal proliferation of T cells. Whenever it becomes small, then you don't have a contribution of anti-tumor immunity. And what we identified is that basically a specific part of the tumor, which is the mesenchymal-like group, but only a subgroup of these mesenchymal-like tumor cells, then contribute to anti-tumor immunity. But on the other hand, if you have the wrong mesenchymal phenotype, you have an immune suppressive phenotype in the tumor. So the next step on our journey to necrosis is now these vascular proliferation and pseudopolysadding areas of the tumor. And now it becomes crowded in these areas because there is like a very high amount of cell death and a high amount of stress signal in these areas. So specifically to look into these stress and metabolic stress, we now used MALDI and combined it with a spatial transcriptomics to identify areas, for example, that we first, oh, it's gone. My time is over. I don't know what happens here. I think it's still, we are back. Okay, so to specifically look if some of these areas that we identify as reactive hypoxia might be different if we look on the metabolic level as well. And there you see these clusters here. I don't know. Probably it's my problem. Okay. So what we have identified is that this reactive hypoxia is almost very similar to the other reactive program, what we call reactive immune. But now if we add MALDI on top to separate these, we can see that basically there's 2 different types of reactive hypoxia programs. So we were curious to see how this is related in space. And interestingly is these core parts, the specific program of hypoxia-associated spots is basically always in the middle of the other hypoxia program. So we assume that there is some kind of an hypoxia core program versus an hypoxia-associated program. And when we looked a little bit more in detail and what is the differences between these 2 programs, one is very highly dependent on glycolysis, whether the other hypoxic program not. And then the next step was to look if we can see on genomic level some kind of alterations. And what we noticed is that basically these programs have a high number of de novo copy number alterations that we can normally not find in other programs or control areas all over the slide. And this is mainly deletions, but it's inferred data again. So this is not DNA data. So we can just basically say that we can assume that potentially there's a higher amount of copy number alterations and genomic instability in areas that are highly affected by hypoxia. So to look a little bit more in an experimental model, we used tissue, and then we cultured the tissue either in hypoxia or in homoxia. And after 6 weeks, we performed DNA methylation to see if there is any kind of an epigenetic dysregulation, and on the other hand, if we have differences in copy number alterations. And surprisingly, yes, we see there's a high amount of copy number alterations, but very noisy. So these very specific copy number alterations that we could identify in our spatial data can be not completely reproduced, but we can see that there is a high amount of de novo copy number alterations in after hypoxia treatment. And on the other hand, also we see a difference in genome methylation. So basically looking at just one promoter of MGMT, which is very important for therapy, we can see that if we have a long-term hypoxia treatment, we almost get every sample to a hypermethylation of these promoter. Finally, we want to summarize now all these results in a little bit some kind of a comic here. So basically, we start with a cell, very healthy, pentose phosphate dependent activation metabolism, and then starts dividing, and finally we came onto a point where the cell switches into a more hypoxic phenotype, and by switching into an hypoxic phenotype you get something like the S phase arrest, so you block cell cycle. And then why it becomes more and more harsh in these areas, the cell underwent de novo mutation, so increased mutational burden, de novo copy number alterations, or also epigenetic reprogramming. And that is for most of the cells, they're ending up in apoptosis, ferroptosis, or necrosis, but only a couple of these cells then might be able to migrate away in less harsh areas of the brain, and then the whole cycle starts again. And that is not only hypoxic stress that might induce these different problems here, but it's also chemotherapy, radiation, and other stress factors that potentially can lead to specific accumulation of genomic instability. So a very easy model to try to get an idea if we see differences between a path to migration and a path to hypoxic activation was that we used vector fields in the spatial transcriptomic data to identify local gradients towards a hypoxic activation. And then we turn it around and said which type of gene set enrichment has an opposing vector to this hypoxic activation. And then we identified that basically migration is always in the opposite direction to hypoxic activation. So that also gives us a hint, at least, that there might be an activation of migratory capacity when it comes to more hypoxic areas. So now we have a lot of insights into the spatial distribution of different subtypes. Now the question is, can we use these for clinical decision, or is this helpful in any way? So I just want to give you a couple of examples why I believe, for example, in the future, we can use these data and also we can make some useful conclusions out of it. And we start with one of an idea that a colleague had a couple of years ago, is to basically set up a model completely in human and to test personalized medicine approaches. And here, this is one example of a BRAF mutated glioblastoma. And the question was, what make different BRAF inhibitor have an impact on tumor growth and differentiation. So basically, we used the cortex of the same patients and then the tumor. We created the slice model I told about already. And then on one side, live imaging. Then we always use electrophysiology just to show that our slides are still alive. And then we said, okay, basically it's expensive, but let's go for a readout using spatial transcriptomics and paired single cell. If this is helpful. And one of the things we noticed is that, yes, we can definitely look in what happens within different treatment conditions with the cell and the differentiation trajectories. And then on the next level is we can also then project these differentiation that we see in single cell into the spatial areas. And we might find different conditions, for example, here, a more enhanced infiltration into cortex or white matter dependent on the therapeutic approach. So spatial also adds a new dimension to really make some kind of a prediction of how a tumor response might work. Another thing is, when it comes to tumor response in GBM, are a lot of failed trials. So over the last years, I think we tried so many different components for treatment, and almost every trial failed. And the question is now, why this? And nobody looked really into the data of these trials. And having now a spatial transcriptomic available that we can also use paraffin slices opens us for like a lot of different samples that we now can look into. And now I think Tenix had also brought up the single cell from paraffin, but at that time only spatial transcriptomic was available from paraffin slides. So we looked in a couple of different trials, and this is, for example, one example of the most recent failed BMS trial was nivolumab. And we just want to see what happens in the tumor before and after treatment. And what we noticed is that basically we see a stronger enhancement of T cells, and we see an infiltration of T cells. So nivolumab is a checkpoint inhibition and should enhance T cell immunity. That was definitely not the case, or basically what we see is, yes, it worked. We see an enhancement of T cell immunity. So you can see this, only a very, very few spots here blink up for T cell, which is like a very strong enhancement if we look at the recurrence setting. But we have a complete reprogramming of the tumor microenvironment. So what we see is basically that the myeloid component also reprogrammed completely into an anti-inflammatory phenotype. And that finally leads there that most of these T cells basically exhausted. And then these patients even do worse after this treatment. And I think spatial transcriptomics can also help to specifically look in treatment failures or early development phase 1 trials to specifically gain some knowledge of the mechanism that might lead to a trial failure. So I want to summarize a little bit. So we have seen that there's a lot of cell types basically that contributed to immune responses of these tumors. We have on one side astrocytes, we have a lot of microglia cells, we have bone-derived cells that we normally separate into a structural immunity part with astrocytes and microglia cells, and a hematopoietic immunity part with bone-derived lymphocytes and macrophages. And all these components are localized in different areas of the tumor and contribute on different levels to anti-tumor immunity. And then we have other components like metabolism or potentially genomic instability that is also co-localized in the tumor and not widely distributed. So it's always very important to look on the spatial level, what kind of molecular alterations you want to look at, and if this is definitely something that holds true for the whole tumor, or if we're just focusing on something that we can see in very small parts of the tumor. So thank you very much. This is my team, and this is especially the spatial team. And we're looking always for new postdocs and especially computational postdocs and PhD students. So when everyone is interested, just give me a mail, and thank you so much. I'm happy to take some questions. Pedro.
The first one concerns cell types. What do you do to separate whatever is between glioma cell and an astrocyte? What is in the middle of the, and how do you manage to separate?
So basically half a year ago, I would just basically say on genetics. Yeah. So I look at the, sorry. So the question was basically how we can separate these different cell types, because sometimes transcriptionally they can be very similar, but how we can see if this is an astrocyte or a tumor cell. And then even I showed that there might be an astrocyte that potentially having some progenitor features that looks like a tumor cell, basically, how we can differentiate this. And I would basically say that half a year ago, we just look at genetics. So basically we look if there is mutations, if there is copy number alterations, it's a tumor cell. If not, it's an astrocyte, even if they have the same transcriptional program. For now, a couple of weeks ago, there is this a paper that's showing that there's a very high integration of astrocytes in the tumor network. So Winkler showed basically that almost 50% of the tumor network or tumor mass potentially are astrocytes, and we can't really distinguish them. And we are also not 100% sure how much of genetic material is potentially shared. So the answer is not as clear as it was a couple of months before. So potentially there can be astrocytes that also have genetic alterations, just basically because it's shared, or like they just shift some copy numbers through the astrocytes. What also was shown is, for example, that they share mitochondria. So some of the mitochondria then travel to the tumor or travel back to the astrocyte. So it's like there's a lot of transfer of genetic material. And I don't know if this is really possible to 100% separate these cell types. Yeah.
So you have spatial platform, and you also have a things that are made by the state which you can overlay in predicting what cell type is where.
Yeah. Yeah.
So it's cell RNA, you have a lot of cell types that you separate the non-malignant astrocytes.
So it's impossible on basically just on transcriptional level, it's impossible. You can't distinguish these, especially when it comes to reactive astrocytes. So basically homostatic astrocytes, sure, there are specific markers. We also believe that there are some proteomic markers like HEPACAM, for example, that is unique in astrocytes. When it comes to the activation, then they share a lot of gene programs together with the tumor cells. So then it's very difficult. The only approach that we normally use is basically we look if they have a copy number alteration. So if they have a gain of 7 and a loss of 10, it's going to be a tumor cell. Otherwise, it's an astrocyte. But as I told, I don't know how much genetic material basically is shared. Yeah, when these astrocytes are completely integrated in tumor networks, potentially they also share some genetic material. But yeah, I think still I would do the same. I would just basically look at the copy number alterations because they are very strong. Yeah, so the gain of 7 is like you can see it. Yeah. Yeah. Chlorodronate is something we use. Yeah. That works not for all areas of the cortex, which is weird. I don't know why. For most of the cortex, it works. But for example, for if you look, so we tested this, for example, in some mice cohorts, is like basically inner cortical areas, like the insular areas, for example, are not affected by these depletion. So we still maintain some microglia cells in these areas of the brain, and they repopulate to the cortex after a couple of days. So this is just I would not say a complete depletion, but a strong reduction of microglia. Yeah. Yeah. Yeah, that is also very, very difficult. So what we have done is basically we have looked at typical programs that we know existing in the brain. For example, radial glia migration programs is something that happens during development. And we thought basically some of these gene programs can be also hijacked by the tumor to migrate. So basically what we have done is we just looked in these vector field analysis in how we can identify gradients in space that follow a stronger increasement of, for example, migrational programs. That is just, it's not really showing you definitely that there might be cells that are migrating because we can't identify them because the resolution is not high enough. And we also have a static view, so we can follow and track the cells. So this is just a measurement to potentially get an idea of if there's opposing activation patterns in space. So pseudotime is something we can't do in spatial transcriptomics. So because pseudotime is basically only possible if you have single cell data or metabolic labeling. RNA velocity also only possible if you have a defined cell type, because as long as you have a mixture of different cells, you not really can play around with pseudotime. Yeah. This we can do, and this we have also done because one of the major reviewer points was that our programs might be unique to specific clones, and it's not really so not unique to some kind of a phenotype that we see in space. And there you can go and look at clonal evolution, and you will also notice that a lot of these clones are basically in a pattern, so they are next to each other, but there's always some spots, for example, in the neighborhood with a distance of up to a millimeter away from the original clone, that also containing a similar copy number alteration signal. But the problem is still the resolution. I don't know if we can completely then infer that that is a migrating cell away from the mother clone. So I'm not really sure if we can make such predictions, but potentially, yes, you see something like this. Yeah.
The different areas necrosis, border. So do you take samples from those different areas to surgery, or you take a sample in which you can see all these areas? And how much do you do this analysis in separate pieces of tissue, or do you do that in the same tissue?
Very great. Yeah, we always try to capture at least 2 or 3 of these regions in one section. But really, sometimes we are completely lucky and capture all of the different parts. But sometimes I don't know, that also might depend on a patient. So sometimes potentially a patient is like having a larger mass, and then you will not be able to cover every different part of the journey. So normally we have like let's say the first 2 stages in one sample, and then we pool it with another sample that have the last 2 stages or something like this. Yeah, it's not only done in samples that have the whole journey.
Migration areas
so the idea is basically HIF-1 alpha and all its related downstream pathways leading to specific programs. And one is, as I told, is a blockade of cell cycle. So basically what we can see whenever we have hypoxia, we don't see there are spots that still expressing mitotic genes. So there's a block of cell cycle. But on the other hand, these cells downstream showed always a downstream activation of any kind of migration genes, extracellular matrix, some kind of these pattern. So that is some of the things we assume, that if you have always a connected program of migration, it might be more likely that these cells also then finally be able to migrate away from these harsh areas, because it would make sense if you say like a cell went into like a stress situation, it can run away and escape. Yeah.
There's a lot of information over the last year or so about cancer and neuroscience. So we've been able to differences depending on where the tumor was located, to be able to look at that.
Yeah, we have looked into this, and still it's not that many samples, so we cannot really cover the whole different areas of the brain. But basically, if it is, so most of the samples are from frontal or temporal lobe tumors, and there's no difference. So basically I have not seen any sex difference or any kind of localization differences. We have tried to look there, but so you always get some significance, but I don't know really if this can lead to any interpretation of the data. So for now, not, but basically, I believe, yes, there might be differences of different areas, but more in a term that the cortex might be different to like infiltrating parts on the corpus callosum, for example. So there might be more stronger differences than just basically looking on different cortex areas. Yeah.
So I'm interested in not studying glioma, but I'm interested in these different areas that you define as the tumor grows, and presumably different distances from heart change. And the question I have is, do you think that these changes in subpopulations are essential for tumor growth? In other words, if you prevented them, that's not tumor growth? or are they just like tissue constants to get this metabolize other things that happen as that?
I have no idea. Great question. The problem is what we don't know is how this architecture is. So what is the developmental steps of these architectures? So basically a tumor starts with a single cell, or let's say a couple of cells, and how we came to these kind of defined architectures that we can see in all patients. And then the next is, if I look at recurrent tumors, basically there is not really a significant change. So basically at the time of recurrence, basically the tumor already has the same architecture, even if it was completely resected. So there seems to be something that this architecture is beneficial for the tumor. But how this architecture and how these different areas are involved, I have no idea. Definitely no idea. Yeah. There need to be some physics involved. So Pedro might have an idea how this is. But
primary and parent.
Yeah. So I looked in a larger set of paired primary and recurrent samples from. That was what I showed, one of the samples from these BMS trial, which tried different therapeutic approaches. And basically, yeah, in some therapies you can see differences, and that is one of the like approaches where you can see definitely something happened in the microenvironment. You see differences between primary and recurrent setting. But for example, in temozolomide, standard stop therapy, we don't see any difference. Most of the samples are almost the same. And you see differences sometimes if you look in specific areas, and that if you look on a bulk level, and I tried this because now there was from Rolf Verhaag, the GLASS consortium was published most recently, and they showed that there is differences in more mesenchymal or more neurophenotype. And on bulk level, yes, we can also see this. But if you then look on the spatial level, then it's just that there are different tissue architectures. You're just looking at different areas of the tumor, and then you get this results of more neuro, more mesenchymal. But if you compare this and normalize by the histology, then there's no difference anymore. Yeah.
Great talk. I'm intrigued by your analysis of doublets. Is that basically spots where you're seeing high expression of different cell types? How are you studying that?
So so the question is this doublet technology, to look at cellular interactions by just using the doublets that normally remove from your single cell data set. So the whole idea came with, there's a technology published as PIC-seq. So PIC-seq, physical interaction cellular sequencing, is basically the idea of sorting cells by either a double positive signaling. So for example, let's say tumor cells green, myeloid cells red, and so you're looking for a double positive, and then mono cells. And so the double positive cells might be interacting cells, and you can separately look at the receptor ligand interaction in these doublets. And we said that basically, if this is possible in a specific sequencing approach, we can also just use the doublets that we have in our data set and look. Yes, of course, there can be technical issues because the cells just basically stick together in your tube after sorting, for example, or they just stick together because they are more likely to stick together. And when we looked into these in these doublets, we found very interesting, because you get your signature from one cell and the signature from the other cell that you seems to be more likely connected because of your spatial data, or because they're expressing receptor ligands and then related downstream and upstream pathways from your receptor ligand interaction. And then interestingly, in the doublets, we can identify just by the signatures of these different cells, which of the doublets these both cell types stick together. And we used these as well then later for when we looked in, there was one study we looked in CAR T-cell interaction. And there also we can prominently see which of the CAR Ts is activated and stick to a tumor cell or which not. So that is really working, I would say. Yeah. Okay. Thank you so much.